while he brings it up, I want to thank the organizers for allowing me to present our work here. It's fun coming back to this conference, kind of seeing how the community has evolved over the years. Now, in my lab, we focus very heavily on developing computational models of metabolism, transcription translation, protein secretion, cell-cell communication, a lot of this sort of stuff, because ultimately we want to understand the molecular basis between how cells interact in their environment. And we want to harness it for producing a number of therapeutic biomolecules and so forth. So I'm going to present some of our work today where we've been able to look into the synthesis of something quite important, which are complex carbohydrates. These end up being bioactive molecules that have a lot of different applications. So first, this is going to be, I'm going to show my acknowledgements. This is a labor of love that was done in part with the collaborators, Lars Bodie at UCSD and many others, but it was really led by some fantastic researchers in my lab, Ben Kellman and Anne Rochelle. And let me actually get my laser pointer. There we go. In case I want to use it. There we go. So it's a long-term project that they worked on where we were able to fully comprehensively enumerate this entire system. This work was primarily funded in part by the Novo Nordisk Foundation and NIGMS. But I also want to point out we have a number of computational postdoc openings. So if any of you want to do a postdoc or want to be a staff scientist, and especially if you're into the modeling and want to apply it to the production of various valuable biomolecules, reach out to me. You might be forced to move into a place like you can see up there in San Diego, where when your nice clothes don't show up at a conference, you can end up showing up like this in shorts and flip-flops. So hopefully some of you are interested in that. Definitely reach out to me. So I'm going to jump into this and talk a little bit about the background. Why do we even care about human milk oligosaccharides? And then after that, I'll go into the modeling work we did. I also have to acknowledge, full disclosure, we started a few companies around our glycobiology stuff, and so it touches upon some of the materials in here. So I'm going to first talk about necrotizing enterocolitis. So necrotizing enterocolitis is a disease many of you probably have not heard about, but it's one of the biggest killers of preterm infants. What happens is you get this bacterial growth and inflammation in the gut, and unfortunately, a lot of children pass away from this. And they don't really know exactly what causes it. They do know you get these bacterial populations blooming, but they don't know really the cause. What they have found, and they've known for a number of years, is that if you feed a preterm infant formula or milk-based products, they have a 6 to 9 times increase in risk in developing this terrible disease. And so when you're thinking about the cause, well, maybe it's something that we could tease apart between the difference between human milk and cow-based products. And so what's in the difference between human milk and cow-based products? Well, when you look at human milk, you have a lot of protein, fat, and lactose in there, as you'd expect. But you also have these complex carbohydrates, these oligosaccharides. Compare that to cow milk, and the distribution is very different. You have these very, very small amounts of the oligosaccharides. So we decided to tease apart, and ask the question: could these oligosaccharides actually be a risk factor or potentially something beneficial to combat necrotizing enterocolitis? So first, I'm going to give it a little background on complex carbohydrates, because many of you probably have not seen these, but oftentimes what we have here is that they're basically these long chains of different sugars. This is one sugar right there. You can see right here, this is lactose. The way these are synthesized, you have complex pathways that elongate these sugars, these carbohydrates by adding more and more different types of sugar. So you can elongate it with a number of them. So I'll just point out this blue circle there, that's glucose. The yellow circle is a galactose. The blue square is an N-galactosamine, and they have a whole panel of different sugars that can be added onto these glycans. They can either be extended or branching. You can add fucose or sialic acids. These are oftentimes terminal sugars. And so what you end up getting is this wide diversity of different sugars, and each one of these have different bioactivities to them. And so there's hundreds of these milk oligosaccharides, and we really don't know what the majority of them do in biology. However, we do know that they modulate your microbiome. Basically, you can change your microbiome composition by feeding these. You can block certain pathogenic bacteria and so forth. They can also be immune modulators. They change the way your immune system acts. And they've also been able to shown correlations with various milk oligosaccharides and brain development. So we do know that they are quite important, and that's why they're actually emerging to be a very large multi-billion-dollar industry now, and synthesizing these and putting them into your favorite formulas. And so what we did is actually went and did a cohort study where we looked at 5 different hospitals and tracked the children that either the preemies that either developed necrotizing enterocolitis or did not. And we found that there was among our cohort, we didn't find any of the clinical covariates could explain whether or not they would develop it or not. However, when we looked at milk oligosaccharide composition, we also didn't see much of a signal. And so what you can see right here, as we increase up here with this number on the side, that shows the severity of necrotizing enterocolitis. Let's call it Bell stage, anywhere from 1 to 3. 3 is very severe. And you don't see with the milk oligosaccharide composition, the overall amount, it really showing any difference between the severe and the controlled children. However, when you break it down and look at individual oligosaccharides, we found one, this diacetyl N-tetrose sugar, and we actually did see a correlation where when you had very low amounts of it, you would oftentimes, that would be associated with very severe necrotizing colitis. And you can actually see this when you look at the over time sampling. The children that had the most severe necrotizing enterocolitis, and some of these passed away, unfortunately, they all had the lowest amount of this sugar. So we did some statistical modeling, and sure enough, we found that the sugar was very much associated with very low amounts of it was associated with a negative outcome. And you could sample it over time, because sometimes you'd have a healthy baby that would have a low amount, but they'd never develop it. But if you sampled over time, after multiple days, and you kept on getting low after low after low, those children would almost always progress towards necrotizing enterocolitis. And so we actually went in, our colleague went in and actually did some animal studies. They actually demonstrated that you could rescue rats with necrotizing enterocolitis with this sugar. But there raises a big question, is why do some women not produce much of this sugar? What's the genetic basis of it? Could you actually also synthesize this either through chemical synthesis or metabolic engineering to be able to produce large amounts to add as a nutrient or a nutraceutical? The problem is we don't actually know the enzymes that actually produce these or how the cell makes them. Even the efforts to make a chemoenzymatic synthesis, they struggle making this sugar in particular. And so that's one thing that we focused on, is we wanted to figure out how is this actually made in the mother. So you could do a simple analysis to say, well, if you see a certain gene upregulated, would you expect, would that be associated with an increased amount of a specific sugar? So there just so happens that we do know one of the enzymes that is critical for making 2FL, which is the one that you find in Similac and so forth that they add. So we do know this enzyme adds this fucose right there, and it also adds it on a few other glycans or a few other oligosaccharides. And so what you can do is you can look at a bunch of mothers, and you would expect that if you're just looking for a correlation, that as you get an increased amount of transcriptional level, mRNA level, you'd expect there to be an increased amount made. But across individual mothers, so each of these lines of different mothers, we actually don't see that correlation. In fact, we see an anti-correlation. And you can look at this and you see this for every single one. So you cannot just take the correlation between expression level of certain enzymes and the amounts of sugars that come out in order to discover all the enzymes. That's because right here, these glycans are made in these complex branching pathways where you'll have a number of enzymes strung together and branching out and to ultimately make your oligosaccharide. So you might have something where you have certain expression states for all these different genes shown as you can see right there, where A has the highest expression and so forth. And then you might, from this system, they would then ultimately find that that A actually is not even detected. This oligosaccharide right there, because it's an intermediate. And so you have this branching and this competition between all the enzymes. And so what happens is if you have another subject where suddenly you have changes in expression, you may end up getting non-intuitive ones where, for example, right here, the D enzyme actually is the lowest, most slowly expressed. It will be the almost the only oligosaccharide you detect in this sort of system, because what's happening is all of the flux gets routed towards D. And so we felt by looking at this that maybe if we mapped out the entire biosynthetic pathway for human milk oligosaccharides, we'd be able to identify all the enzymes in there. So that was a work that was led by as I mentioned, by Anne Rochelle and Ben Kelman, pictured there. And so what you can do is you can start to, on paper, start drawing out these biosynthetic pathways. So you can start with your lactose right there, and you ultimately want to make all these other oligosaccharides. You can start drawing the lines between them for single monosaccharide additions and so forth. But one thing you'll notice is that you could actually take other precursors and draw maps, draw individuals, multiple steps to get to a number of them. So there's a lot of different network topologies you could have there. So we wanted to search through these to identify what is the order of biosynthetic steps, and then what are the enzymes used to produce it. And so with that, we took all possible enzymatic additions and mapped that out into a very large network. It ended up being almost 10,000 reactions, and suggested that you'd have about 3,000 milk oligosaccharides that could be used to make the 16 most abundant ones. Now, when mapping out this network, what you'll do is you'll find that there's a number of them that, for the terminal oligosaccharides, that are not actually ever detected. And so you can go in, and to get a more refined network, you can go back and remove a lot of the intermediates that are never really used for the measured oligosaccharides. And so what that gave us is we ended up with a candidate pathway that had 221 reactions to make 101 milk oligosaccharides that were essential for making the 16 most abundant ones. Again, there's a lot of different routes here that could go to make each one of these measured ones. And so what we wanted to do to find the most likely usage of this pathway, you could go through and you could use, you could basically take all these different routes to get to all the same oligosaccharides. So maybe there's certain reactions there that aren't real, that aren't really preferred by the enzymes. And those are just two examples of different flux states you could have to get to all 16 oligosaccharides. So what we did is we ran through and used a mixed integer linear programming to search for all the minimal network topologies, and found that there were only 45 million. And so we didn't really want to manually go through all those. And so we wanted to do some further analysis to identify what is the most likely topologies. And if you could do this, then you could identify the enzymes that were most likely to be involved. You could identify their specificities so that you could then potentially use these in metabolic engineering, or you could actually go to answer these questions about metabolic trait or enzymatic traits and genetic variants that ultimately influence the milk oligosaccharide profile and thus the infant development. So what we did is we identified a couple dozen enzymes that were annotated as potentially having catalytic activities on glycans that could actually, in some cases, actually catalyze similar reactions. And we mapped these onto this network. We also used a modeling approach here where for each of the subjects, for each woman, we were able to measure the amount of milk oligosaccharides that were produced and use a flux balance analysis approach to be able to simulate the fluxes through all the individual reactions. This was done on each of the 45 million pathway topologies. And we did this for every subject. And then what we did is we took the gene expression data from the mammary glands, from the mammary epithelial cells in those women, to look for correlations of gene expression with metabolic flux, not the abundance of the glycans, but rather the metabolic flux. And so you'd see right here, in some cases, you would have a good enzymatic correlation with the flux. In some cases, you'd have a poor one. And we'd then go through, and for each of these, you'd have multiple candidate enzymes at each link. And so you could identify the enzyme that was most giving you the best score, so that you could then get the best pathway topology and the best enzymatic composition score for each of these. And so we did this on 2 independent data sets from in a total of 11 women taken at multiple time points in postpartum. And we were able to, from there, identify a subset of model topologies and candidate enzymes that would give you the best score. And fortuitously, they actually overlap. They actually gave us very significant. There was a significant overlap when we get the best enzyme batch and network topology in one data set. We'd actually see that in the other data set too. So with that, we took these best overlapping ones, and this is roughly what the metabolic network topology looks like for all of these oligosaccharides. And so you can't really see very well right here, but you can see there's different enzyme different types of enzymatic reactions at each individual step. But we want to know, what are the enzymes catalyzing all these? And so going back to that initial story where I showed that you could not predict that even the known one, FUT2, which catalyzes fucosylation of lactose, you couldn't even predict that based off of just the correlations between its abundance and the transcript abundance. Well, once you account for the fluxes and so forth and look at those correlations, looking at these would be all the steps that it would be needed for synthesizing it, we actually get that FUT2 is the only enzyme that gives a good fit on that. So we were able to accurately predict the known one. We were able to then go through on all the 10 different types of enzymatic linkages that are found on these milk oligosaccharides. And we were able to find very strong candidates for every single one of them. And I'll mention, since this is more about the modeling, we did go in and experimentally validate a number of them, and they all came out as working. So in summary, individual glycans are very important. These oligosaccharides are very important for infant health and adult health too. But these glycans are part of a complex system, and you can really only tease this apart by building out the networks, simulating the fluxes through them, and drawing correlations between the different omics data types to be able to identify the enzyme. So the technique here could probably be applied to many other different complex biosynthetic systems, but this worked particularly well for us in glycosylation. So with that, I'll take any questions.
Thank you for this wonderful talk. There are questions.
Okay.
Thank you for a really interesting talk. I just want to ask for your opinions or knowledge on the roles of the placenta during pregnancy would play in the secretion or the production of MHOs.
That's a really good question. So I'm not a big expert in that, but I know there's some crosstalk between hormones between the two. And I think that, and there is a lot of research where people have been looking into actually some of the hormones and so forth, how they might actually influence necrotizing or colitis and others. But it's definitely something that we need to look more into to identify the regulators of these enzymes. That's actually something we did do in the paper. We did identify a number of transcriptional regulators that were potentially involved in this also. Good question, thanks.
We can hardly hear you.
What was that? Yeah. So the question is, she has a question about one of the topologies. Was it? Have you seen it yet? Okay. So basically what this is like, you can think of it, these are all possible pathways you can take to make the 12. It's kind of hard to see here, but in here we have the ones that are in red are going to be the ones that are actually experimentally measured oligosaccharides. And to get to those 16, you can take different routes. And so these are basically minimal networks. So you're able to trim out all of the little ones, all the reactions in gray, and still get to the 16 measured oligosaccharides. So that's where we had 45 million different of these different topologies that you could have, and while still getting to the 16 measured oligosaccharides.